Hi, good morning, good afternoon, or good evening, everyone. So it's our great pleasure to welcome you, Miko Seppänen and myself, to this live session on registry and working genetic party session. So altogether, we will listen to 5 speakers. And the first one, Julia Pazmandi, a PhD student who's working at Ludwig Boltzmann Institute for Rare and Undiagnosed Disease in Vienna, in Austria. She will present us an update on human phenotype ontology. So, Julia, please, you're on stage.
Thank you very much for the introduction. So yes, as Anna said, I will be talking about human phenotype ontology for inborn errors of immunity. And I have nothing to disclose. Human phenotype ontology provides a standardized vocabulary for human phenotypes. It currently has over 13,000 terms that are ordered in a hierarchical structure, and these terms are linked to disease databases. There is an effort on its way to translate the original English terms to other languages, and there's also a plain language translation available. Here on the right-hand side, you can see like a very simplified example branch of the HBO tree. On the very top, there are general terms, and as we go down into the hierarchy or to the bottom, the terms get more specific. There are many benefits of adapting HPO as the unified nomenclature of patient phenotyping. It allows for efficient data exchange between clinicians and researchers. It provides a unified nomenclature for databases, data sharing platforms, or publications. It allows for very accurate patient-to-disease or patient-to-patient similarity measures. And it also facilitates variant prediction and gene discovery through tools such as Exomizer, which use human phenotype ontology to prioritize variants from exome sequencing. Unfortunately, HPO is not widely used in the inborn errors of immunity community. And the reason for this is HPOs are incomplete for rare immune disorders. And there are a few problems. A lot of the times, terms are missing from the hierarchy or the structure of the hierarchy is incorrect. So here on the left-hand side, you can see an example branch of the HPO tree, abnormality of immune system physiology to be more exact. And if you look at the first daughter term, abnormal lymphocyte physiology, just by looking at this term, you can immediately tell that at least one term, abnormality of B-cell physiology, is missing, for example. But you can find missing terms anywhere. So here at the immunodeficiency, you can see that B-cell deficiency is missing. Furthermore, if you look at the terms that are in the same level of the hierarchy, so here on the left-hand side with the different colors, you can see that there are very different and diverse terms at the same level of the hierarchy. So it's not completely optimal. Furthermore, diseases are often not annotated with HPO terms, or terms are missing, or the terms that annotate diseases are not accurate. So here you can see the official IUIS description of ADAM17 deficiency. And if you query for the same ADAM17 deficiency in HPO, this is the list of terms that you find annotating ADAM17 deficiency. And just by comparing the IUIS description to what we find in HPO, we can immediately tell that there are terms missing. So it looks like the HPO needs some completion and some restructuring for rare immune diseases. And in order to do this, we have started an initiative in 2018, spearheaded by the LBI Root, the ERN Rita, and ESID. We had 3 in-person meetings so far and many, many remote ones. And currently we have over 30 members across 9 different countries, among them clinicians, geneticists, immunologists, and computational biologists, and some of these members are actually here today. And we have a remote working structure that is quite functional. We have 4 working groups addressing different types of inborn errors of immunity. And with these 4 working groups, we set out to restructure and complete the HPO tree and to reannotate inborn errors of immunity with HPO terms. We have chosen a very hands-on branch-by-branch approach for our restructuring effort, and that mainly happened at our in-person meetings and resulted in notes such as the one you see here. And we devised an ontology-guided machine learning approach coupled with an expert review to reannotate diseases with HPO terms. So very briefly, the basis of this approach was an expert-curated list of publications on which we have used a text miner to extract phenotype terms and translate them to HPO terms that were sent to a group of clinicians to review, and multiple experts reviewed a disease. So it was a double review process. So as I just mentioned, we have used a very hands-on approach to restructure the tree, and we had a lot of notes like this, and these were translated into a unifying format. And here, I just wanted to show you a few of the example branches that we have revised. The new introduced HPO terms are highlighted in green, and the repositioned terms you can see in yellow. So in summary, so far we have kind of dipped our toes into 4 main branches of the HPO tree and requested over 200 changes, among them 57 completely new HPO terms. And this effort is still ongoing. We used this ontology-guided machine learning approach to reannotate diseases with HPO terms, and so far we have completely reannotated 72 inborn errors of immunity. And we have seen that after reannotation, the number of available HPO terms per disease has increased significantly. So we saw a 4.7-fold gain in number of HPO terms available per disease. So here on this plot on the upper left corner, every single dot represents a disease. And not only did we see an increase in the number of HPO terms, but also the available information content per disease increased after reannotation. So if we look at now an HPO annotation of like an average disease in the 72 reannotated diseases, what we see is that the majority of the terms come from the text mining, so over 70% of terms. There's also an about 8.6% overlap between the already existing terms with the text mining terms. And we have also asked experts to suggest additional terms or additional phenotypic descriptions if they felt like neither the existing or the text mine terms really accurately describe the phenotypic spectrum of the diseases. So hopefully I could till now convince you that indeed we have gained a lot of new HPO terms per disease, and also the information content increased. But of course, our main goal is to create a complete HPO annotation and description that is good enough to use in a clinical setting, is good enough to use as a diagnostic aid, and is very accurate in patient disease similarity matching. Unfortunately, because we have not finished reannotating all of the diseases yet in the inborn errors of immunity, I cannot systematically assess how well the reannotation has improved the similarity and matching. But I brought you 2 examples now that illustrate that we are kind of going into the right direction. So here's patient A, who goes into the clinic with the following clinical description, and a very expert immunologist would immediately diagnose this patient with common variable immunodeficiency. What we can do as non-expert clinicians with HPO is identify the phenotype-like terms, translate them to HPO terms, and compute the HPO ontology-based similarity of these phenotype terms to CVID. And what we see now is that actually the matching accuracy of these patient phenotype terms to CVID increased after the reannotation of the 72 diseases. Patient 2 goes into the pediatrician with the following description, and this pediatrician has a lot of expert immunologist friends, and they quickly come to a diagnosis of TRAPS. Well, as non-expert immunologists, what we can do with HPO is identify the phenotype terms, translate them to HPO terms, and compute the HPO-based similarity of this set of terms to TRAPS. And again, what we observe is after a reannotation effort, the matching accuracy of patient phenotypes to TRAPS increased. We're also wondering that beyond patient to disease similarity matching, how did disease similarities change after our reannotation effort? So here we used, again, the HBO ontology-based similarity measures between all diseases that we have reannotated before and after reannotation. So this is kind of the similarity wheel that we get before reannotation. And here, every single leaf is a disease. They're identified by their OMIM identifiers, and they're colored by which clinical subgroup they belong to in the IUAS classification. And what we see after reannotation, that indeed those diseases which belong to the same clinical subcategory, they are more similar phenotypically based on HBO after reannotation as compared to before. So it looks like we are going into the right direction. Of course, we are not done yet, and we are sort of only getting started. But I think already we achieved quite a substantial gain in overall annotation and information content for the diseases and the 3 parts that we have reannotated. And our immediate goals are to finish review and reannotation of all inborn errors of immunity, and to aid this, to facilitate this, we would like to spread the word with talking at conferences such as this, also to secure collaborations and funding to do this work further. Our dream is, of course, that HBRs are widely used in both internal and external databases as well as publications, and that HBRs are adapted in clinical care. And with this, we want to take a big step forward towards data-driven medicine for inborn errors of immunity. I have a lot of acknowledgements to make, or people to acknowledge. First and foremost, I would like to acknowledge everyone involved in the initiative, every single person. It would fill a whole slide with just names if I had mentioned everyone, especially the 3 people on the right, Matthias, Marielle, and Khan, who are key people in this initiative. And I'm a PhD student, and I have 2 supervisors, Khan Bostuk and Jörg Mentje, and I would like to thank them both. Thank you very much for your attention.
Thank you, Julia. That was exciting. Thank you, Julia. That was exciting. Next, we will have Markus Seidel, Professor of Pediatrics at the Medical University of Graz, Austria, who's trained in pediatric hem-onc and has been a long time active in the ESID Registry and a progressive researcher in the field, to suggest whether we would benefit from implementing HBO into the ESID Registry. Thank you, Markus.
Yeah, good morning, everybody. These are my disclosures. No, it doesn't. Okay, so actually starting with the initiative Julia presented right now, together with the ESID Genetics and Working Party and ERN Rita, I immediately thought that implementing the HBO terms in the ESID registry would also be very beneficial. For example, in the part of the working definitions for the clinical diagnosis that has been an ESID registry working party initiative since many years. You probably know this catalog of about 100 diseases, many of which are not genetically defined, and if you want to enter a patient into the registry without a genetic diagnosis, you have to fulfill these working definitions for the clinical diagnosis. If you, for example, look up autoimmune lymphoproliferative syndrome, you would get to this page and find OMIM disease codes here, the IUIS category, the OMIM gene code, the ORFA disease code, and most importantly, the clinical criteria for a probable diagnosis, or what we call them the working definitions, that have been established by many experts of the individual diseases and that have been peer-reviewed by other experts. And now what Lucas Gasteiger did as a medical diploma thesis project is that with the help of Peter Robinson and a lot of computer algorithm tools, we added HBO terms that were already existing. So we know that the number is increasing, like Julia explained just now. So if you now click on this button here, you will be linked to most of the times about between 1 and 4 page form within the same document that tells you about the very frequent HBO phenotypes, HBO encoded phenotypes that are found in this disease, the frequent ones, the less frequent ones, and the infrequent ones. And so this is just basically an example how we could add HBO terms to the diseases that are already in this catalog. And there are ongoing initiatives like the GAIN registry that uses HBO terms, the immune deficiency and dysregulation score that's being set up within the ESID registry to prospectively monitor disease activity, immune dysregulatory disorders, and probably other registry projects will follow. So the potential extension now of the use of HBO terms in the ESID registry would be to record every fulfilled criterion in a patient, first at the initial diagnosis and the first entry, and then every year at the follow-up. For example, by clicking on the clinical term or on the lab abnormality, this would automatically be added with the HBO code. And this would have 2 different consequences. First, we would have searchable terms and match to be able to match them with omics, whatever, genomics, transcriptomics, whatever. And we could record the longitudinal course of the patient. For example, it could be the case that after 2 years of follow-up, a few more criteria would be fulfilled. But on the downside would be that it's very difficult for documentarists, and each entry is more work, and it's technically quite difficult to implement this because as the registry is set up right now, it is not really designed to prospectively monitor the clinical phenotype and the course of the disease of every patient over time, only in specific sub-projects. The other potential extension would be to use what is already there. For example, to add terms here. This could maybe be done also in the background, but this would probably be a small compromise and only lead to a limited gain of information. So, in summary, the potential gains and pains, as I see them, is that adding the HBO terms in the ESID registry would probably make the search across registries and platforms more compatible. The definition of inborn errors of immunity phenotypes will improve. The known disease-causing genotypes will be described better, and the gain of knowledge output of the registry, especially through the use of machine-assisted tools, would be probably really big. But it would need a step-by-step restructuring of registry data. It would need an integration with the HBO community and the servers at the Jackson Labs. It would need to implement all the new HBO terms to existing fields and parameters and a continuous curation. And the creation of a longitudinal monitoring would be probably very labor intensive, and not only quantitatively but also qualitatively. If this is prioritized by the ESID board, it's probably be an expensive journey, and it's potentially difficult to be carried out by the documentarists alone. So the quality it's a matter that we should discuss. Do we really want that? So thanks for your attention, and happy to discuss these points later. Thank you.
Thank you, Marcus. It was a great, short and great presentation. So Mikko, you take the... Mikko?
For some reason, my laptop is frozen. Oh, no, okay. There's a great lack of time before you can jump in. Okay, so the next speaker will be Andrés Caballero, who has finished his master's degree in molecular biology in the Autonomous University of Madrid, and his PhD in University of Tübingen, and currently he's at the CCI-IFI University of Freiburg and works as a researcher mostly on genetics. Please, Andrés.
Sorry. So, thank you for the introduction. I'm happy to present today our new database called GEMMA. So first, just to mention that 200 of our exomes were sequenced by Novartis as part of a research collaboration. So I'll start off with a couple of examples of situations that you may encounter while analyzing genetic data. So the first one is when you have your pre-filter list of candidate variants. You will probably come across the situation where you want to know what is known about the gene or about that specific variant. Is it pathogenic? Is it what's the frequency in the population? What about prediction scores, functional evidence? What do others say about in all the databases such as OMIM on ClinVar? So I just picked this. Wait, I'm not seeing my screen. No? Okay. Sorry. So, as I was saying, this is what I have to disclose, and these are the two examples that you might encounter while analyzing genetic data. So, the first situation, you have your pre-filter list of candidate variants, and you would ask yourself this type of question. So, what is known about the gene? What is known about the variant? So in this case, I picked this example, OIS1, because, for instance, it is currently not registered in OMIM as a gene associated with inborn errors of immunity. And unless you know the literature or you have a specialized database such as this one, you would probably miss this mutation. So a second example would be when a novel gene is reported in the literature. So you want to know probably how many families are reported, what is their clinical phenotype, whether I can compare those cases to my patient. This could be easily answered if this data was already in our database. You could easily query and find out. Or more importantly, you would probably want to know if in some of your exomes, you could have also additional patients carrying mutations in this gene, maybe the same mutations, maybe similar mutations, and you could easily do a query in your database. So basically, to answer these questions, we wanted to create this database because by that database in our sequencing data, so that means every single variant identified in the patient, all the metadata associated with it, and by integrating this with additional data sets, so clinical information, functional evidence from internal assays that we perform in our group, also from external groups or collaborators, or even published in the research literature, so we can pull together all this information using also additional external resources such as OMIM, ClinVar, GenoMat, Ensemble. And what's important is also to integrate it with ontology. So as Julia was introducing in her first talk, it is very important that every So all of our patients are assigned to this specific universal terms, so that then we can cross-reference with different databases and do better annotations. So we integrated many ontologies, including the HPO, and probably we would need to update now the HPO with the work that Julia was presenting before. That would be very beneficial for us. In addition, we also have included the microbiome data from our 16S RNA sequencing, including fecal immunoglobulin values and curated clinical data extracted from detailed questionnaires. So this is a snapshot of the type of data you can currently find in our database. So we have over 6,500 individuals. That includes patients, controls, family members. And we have genotype data on at least 2,500 individuals. That includes about 700 exomes and 1,700 targeted gene panel results. But also we have almost 500 individuals for whom we have 16S RNA sequencing data. But in addition, we do this clinical or genetic curation of variants that we identify in our patients. We also curate the genes, but in addition, we also link these genes or papers with genes or variants with the papers in which they were published. So sometimes if you go to public resources that do automatic annotations, you may miss many important papers. So it's important to do this manual step to not miss important publications. And we also have at least 1,300 individuals in our database have been published, and you can find them here with the code that was originally published in that paper. So how is this database accessible right now? At the moment, it's just internal for the CCI here in Freiburg. But of course, we want to make this public at some point. So we have currently more than 30 active users that access through this basic graphical user interface called GEMMADB. It is built on PHP. But however, if you're a more advanced user, you could use programming languages like R to do your queries and analysis, create your figures. Of course, you could also use, for instance, Python. But so I will just show you today like a snapshot of what this graphical user interface looks like. So as you can see here, we have several different modules where you can have detailed information about clinical terms, diseases, all matched with the different ontologies, information about genes, papers, physicians that send us samples. But I will jump directly to the subjects module because of the time. And within subjects, we can find different submodules. So starting by the clinical, we have information about the main diagnosis, main genetic or clinical diagnosis of this patient with you could have age of onset, age at first manifestation, age at diagnosis. If you scroll down, you could have information about additional manifestations or diagnosis assigned to this patient. Again, this is also longitudinal, so you could assign the same thing over time. Maybe at age 10 it was present, but it was not present at age 15, so you could do this follow-up. But you can also, as you can see here in the list, you have the standard term and the alternative terms. But if you click on the term, you would be redirected to the description, the full description of the term, where you can find also the ontological classification and their place in the hierarchy. So if you go now to the navigate to the genetic module, you will find information in this first list above, information about how many times this patient has been sequenced or screened by different methods, including Sanger sequencing, targeted gene panels, whole exome, whole genome. So you have here information about the different runs or studies that were performed with this patient. And you could also have immediate access to the latest report or the reports of these studies. If you scroll down, you would have a list, a pre-filtered list of candidate variants identified in this patient. And here you have just a few attributes that you could see of the variant. But if you click on the link you have access to the full description of this variant, meaning specific comment, manually curated. You also have information about 50 different annotations, including prediction scores, frequency data, splicing scores, information about others, so other individuals carrying this mutation, papers, other transcripts. So just to show you quickly, here you could have a look at the different carriers of this mutation in our database. And of course, the benefit is that you can see immediately their clinical and genetic diagnosis, what was the data from the sequencing, so what was the depth and the alternative read count of this variant in that patient. You could also have access to papers in which this variant was published, but also papers where the patient was published. And if you can have immediate access to the paper and have access to the PDF and have a look yourself at the publication. Finally, you can also see what is the effect of this same mutation on the different gene transcripts. So for this particular example, it's actually the same, but as you may know, for many cases, the cDNA or protein position changes according to the transcript. So now if we navigate to the lab module, the lab submodule, we can find detailed information about blood cell counts, so about different lymphocyte counts. You can see, again, you can do a longitudinal follow-up over time. How were these values up and down during the measurements of this patient? If you scroll down, you also have information about immunoglobulin levels. So all this lab information we get it from our clinical databases at the university hospital. And finally, so if we move to the microbiome module, again, as with the genetic, we have information about the different sequencing runs, different studies that were performed of this patient at different time points. And if you scroll down, you can have a look at the different bacterial species that were identified in this patient, how much they were enriched, what's their relative abundance. Of course, this is just a prototype, and we would like to have a more graphical way of viewing this data, but this would be happening in the near future. So finally, if you go to the personal module, you probably, so you will have access to the family. And if you click on the family, you have all the family members associated with this patient. So also information where they were published, who's the father and who's the mother. And what is important is that having this information allows you to create automatically a family pedigree that you can access here. You can download as a PDF, and you could basically do in a semi-automatic way build this pedigree without any errors. So, and this works with very complex families. We tested with more than 50 individuals, and it works pretty well. So finally, just a quick example of what an advanced user could do by directly querying the database with a program like R. In this example, let's say you want to compare how much or how much more frequent is variance in a specific gene, such as TACI, in which so it's a gene with incomplete pentas, or maybe consider a risk factor, but you could compare what's the frequency of certain specific mutations in the gene in comparison with external databases such as GEMMADB. So what we could see here is that, for instance, the 104, one of the most common mutations in TACI, is enriched in our cohort, in our PID cohort. It is 25 more times common in our cohort than in external databases. So supporting the role of this variant in disease. So just to sum up, I hope I could briefly show you that with GEMMA, we intend to do a specialized database focused on immunology with a strong genetic background. You can store all your sequencing data, also including metadata, and it's a very efficient way of storing this data. As you know, FASTQ files are large, and probably with this approach, you could get rid of them. I mean, at some point, because the storage is very, very low in this setting. And in addition, what is important is that we can combine this data with other clinical data, lab values, functional data, that this is something you could not do with other databases such as ClinVar or OMIN, which are great, but they're not specialized on immunology. So just as an outlook, what we want to do in the future is to perhaps do a more interactive filtering tool from our web application. And we want to offer automatic analysis starting from FASTQ files that you could send us. And so your data could become part of this database. Of course, we need to go through an ethics vote to open to external collaborators. And by in order to do that, we'll first try to do it with our collaborators in Hanover as part of our RESIST consortium. And finally, we want to make it available internationally. So with this, I'd like to thank Professor Grimbacher and Proietti for their help and support with this project, and also especially Matej Krauts, but also the rest of my colleagues in the group who helped and contribute to entering the data in this database.
Hi, thank you, Andres, for this very nice presentation. So we are running late. I'm presenting the last speaker, Frank Rapaport, with the postdoc working at the Seidel Laboratory of Immunology at Rockefeller University and in New York. And Frank, let's go. And sorry for the time.
So let me share my screen. So hello everyone. I'm going to talk today about the negative selection at inborn genes that underlie inborn error of immunity. So first of all, I want to thank the organizer for giving me this opportunity to talk today, and I have nothing to disclose. So the idea, like the definition of negative selection is that it is basically the natural process of elimination of the unfit. The idea that when individuals carry a variant that confer them a loss of fitness, then this individual are going to have less offspring, and this variant will decrease frequency within the population and will eventually disappear. So there being this variant that confer a loss of fitness are being selectively purged from the population, and it's this natural process that we call negative selection. And we say that the gene is under negative selection if its deleterious mutation are being selectively removed. The idea behind this project is that we wanted to measure and to see what were how was the negative selection behaving at inborn error of immunity loci. And the reason why we wanted to do that is not only natural scientific curiosity, but also because we wanted to see how we could use natural selection, negative selection, as a tool in order to formulate hypotheses when looking for new candidate variant and candidate genes. So there are 363 genes in the latest inborn error of immunity classification for which we were able to obtain negative selection score. More than 2/3 of those are underlie autosomal recessive disease. 60 or so of those underlie autosomal dominant disease. 30 of those underlie disease that can be either recessive or dominant, underlie both recessive and dominant disease. And there are 20 genes that underlie X-linked disease, with 19 of those being X-linked recessive and only one WAS, which is X-linked dominant. So when you look at negative selection score, a gene-level metric that aim to measure negative selection, they all try to do the same thing on the paper, meaning that they try to compare the observed deleterious rate within a gene to the expected deleterious rate. If the observed deleterious rate is lower than the expected deleterious rate, then this gene has less deleterious mutation that you would expect by chance, so this gene is under negative selection. However, if you look at them precisely, each one of them, so we found 7 in the literature for which we were able to compute them for at least 75% of the genome. It turns out that these metrics are poorly correlated. So there are 3 different groups. One of them is Lovetul and Evotol, which are a negative selection score that use protein constraint. Another one is LUF and PLI that put some emphasis into the difference between heterozygote and homozygote. And then there's the other one, which are ERVIS, the F parameter from SNP, and SIS. And so we decided to use a consensus-based approach, and we constructed a new score, which we call CONS, for consensus negative selection. So like the CONS. It's a score that's, so on the left-hand side, I'm showing you the distribution of this score on the human genome. It's a score that has a mean of 0, it centers on 0, and a standard deviation of 1. And a negative value of this score is correlated with a stronger negative selection. So we looked at how this score behaved on H-OMIM, which is a hand-curated version of OMIM that only contains highly penetrant genes, and that's what I'm showing on the right-hand side of this slide. And we compared it to random autosomal gene, AB for autosomal background, and random genes on X. And by random, I mean genes that are not known to cause any disease or known to be essential. And what you can see is that genes that underlie autosomal recessive disease in this hand-curated, highly penetrant version of OMIM are not under more selection than a random gene on the autosome, while the one that underlie autosomal dominant disease are under more selection, and the ones that are X-linked are under more selection than a gene on the background. So what happens for the gene that underlie inborn error of immunity? And we can see the same thing for gene that underlie inborn error of immunity. So on the left-hand side of this slide, you have AB for autosomal background genes, so all the genes that are on autosomes, and do not underlie any disease, nor are known to be essential. Then you have the genes that underlie autosomal recessive inborn error of immunity, then the gene that underlie autosomal dominant inborn error of immunity, and finally the genes that underlie both recessive and dominant inborn error of immunity. And what you can see is that the one that underlie autosomal recessive inborn error of immunity are not under more selection than a random gene, while the one that underlie autosomal dominant form of the disease are significantly under more selection, 10 to the minus 9, the p value is of 10 to the minus 9, and the one that underlie both recessive and dominant form of the disease are also under significantly more selection than random genes, the autosomes. If we look at what happened on the chromosome X, the chromosome X is itself under more selection than the autosomes, and that's fairly logical because since half of the humanity only carries one copy of this chromosome, so any deleterious mutation on this chromosome is going to get purged out of the population faster. And that's what you can see with the box in the middle that is the background gene that are on X, and they're under significantly more negative selection than the one that are on the autosome on the left-hand side. But the gene that underlie X-linked inborn error of immunity are under even more negative selection than random gene on chromosome X. And the p-value is very strong, to the order of 10 to the minus 5. There's one exception to this rule, which is the gene CSF2 array, which actually has a positive value of cons, meaning that it's under less selection than the mean of cons. And the reason for that is because even though it's an X-linked gene and associated with a very severe disease, congenital pulmonary alveolar proteinosis, it's actually in the pseudo-autosomal region of the X chromosome, meaning that females have 2 copies of the gene since they have 2 X chromosomes, but males also have 2 copies of this gene, one on the chromosome X and one on the chromosome Y. And this gene is actually recessive, meaning that the males that present the disease also have a Y version of the gene that's mutated. So this gene actually acts more like an autosomal recessive gene than an X-linked gene. We also wanted to see if we could see difference between disease that are more severe. So we classified every inborn error of immunity within 2 categories. The first one are severe disease that are early onset and highly penetrant in pink. And the other one are all the other disease, so either late onset disease or lowly penetrant disease or disease that are much milder in blue. And the first thing that we saw is that most of the known autosomal recessive inborn error of immunity or X-linked inborn error of immunity are actually what we early onset and highly penetrant and severe, while most of the dominant one are not. And this is very significant with the p-value of the order to 10 to the minus 17. And this can be explained by different evolutionary process, meaning that diseases that are very severe tend to evolve and become recessive with time. But if we look at how that impacts the negative selection, actually what happens is that the severity, the high loss of fitness associated with the disease doesn't impact the selection, or we don't see an impact on the selection for genes that underlie dominant disease or both recessive and dominant form of the disease, or even X-linked disease. But we see a very strong impact on recessive genes, on genes that underlie recessive disease. To the point that even though the gene that underlie recessive disease as a group are not under more selection than random autosomal genes, the one that underlie early onset highly penetrant disease are under significantly more selection than random genes with the p-value at the order of 10 to the minus 3. We also wanted to see if the mechanism of dominance would impact the negative selection. So genes that underlie autosomal dominant inborn error of immunity can do it through three mechanisms. They can do it either through gain of function, or through loss of function by haploinsufficiency, or through loss of function by negative dominance. A gene that underlies loss of function by haploinsufficiency means that the mutant protein is non-functional, and there's only half of the level of the protein that's functional, the wild type one, and that's not enough for the protein to be efficient, and that causes a disease. While the one that underlie negative dominance, that act by negative dominance, are actually a gene where the mutant protein perturbates directly the function of the wild type protein. And what we see if we look at negative selection is that the gain of function, the genes that underlie disease that act by gain of function, are under significantly less negative selection. They have a higher value for counts than the one that underlie autosomal dominant disease by loss of function with a p-value of the order 10 to the minus 4. And within the one that underlie disease by loss of function, the haploinsufficient genes are under significantly more selection than the one that underlie a disease by negative dominance. And one of the hypotheses for which that happens is the fact that if you have a gene that underlies disease by haploinsufficiency, then any loss of function mutation may cause the disease, while for a gene that underlies negative dominance, only a handful of loss of function mutation may cause the disease. We wanted to see if this was these were results that were specific to inborn error of immunities, or if we could replicate these results into another similar group of genes that underlie severe and early onset inborn errors. So we collected from our collaborator, Joe Gleason, a list of genes that underlie inborn error of neurodevelopment. So there are more genes, more than 1,000 of genes. Like for inborn error of immunity, most of them underlie autosomal recessive inborn error of neurodevelopment. A smaller number, 1/3 or so, underlie autosomal dominant form of the disease. A smaller number underlie genes that can be underlie both recessive and dominant form of the disease. And another part underlie X-linked disease, with the interest that we have more X-linked dominant disease inborn error of neurodevelopment. And there's a few of those X-linked disease, we don't know if they're recessive and dominant, and I label them as X unknown. So like for inborn error of immunity, the genes that underlie autosomal recessive inborn error of neurodevelopment are not under more pressure than the autosomal background one, but the one that underlie autosomal dominant form of the disease are significantly, very significantly, and the one that underlie both recessive and dominant form of the disease also are. The p-value are stronger than for inborn error, that for gene that underlie inborn error of immunity, but that's also due to the fact that we have more genes in each of these categories. On the X chromosome, there's virtually no difference between the gene that underlie X dominant and the gene that underlie X recessive form of the disease. But taken together, all the X-linked form of the disease are under significantly more pressure than the random background gene on chromosome X. The inborn error of neurodevelopment community doesn't label their genes if they are gain of function or loss of function, but they label them as haploinsufficient and not haploinsufficient. So we are also able to reproduce these results that the haploinsufficient genes are under significantly more pressure than the autosomal dominant, not haploinsufficient genes. So finally, another thing that we wanted to do is to see if we could use negative selection in a supervised model to predict if a gene is likely to underlie a dominant or recessive form of the disease. So once again, as an idea to use this as an hypothesis generating tool. So what we did is that we complemented Domino, which is a supervised score that already does that, with negative selection score. And then we use as a training set the genes that underlie the inborn error neurodevelopment, the one that underlie inborn error of immunity, and this hand-curated version of HOMIM, H-HOMIM, that only includes highly penetrant genes. And we kept the genes that were strictly underlying AD disease or strictly underlying AR disease. We trained a random forest model, and we constructed this new score that we call SCONS, for supervised CONS, as the probability, according to this classifier, that a given gene underlies a recessive disease rather than a dominant disease. And as I said, so the name of this new score is called SCONS. And the first thing is that this score performs slightly better at this task than Domino by itself. So introducing this negative selection score does indeed improve the performance of this classifier. And you can see that on the left-hand side with the area under the precision recall curve, where we do a little bit better. Domino itself doing better than the unsupervised version of the score, CONS, that I presented before. And on the right-hand side, I'm showing you this distribution of the score on the gene underlying inborn error of immunity, separated by the mode of inheritance. And what you can see that most, like the vast majority of the gene underlying autosomal recessive inborn error of immunity have a very high value for SCONS. So they're predicted to underlie AR disease, autosomal recessive disease. But the one that underlie autosomal dominant disease, actually there's two groups, one that are well predicted and a smaller group that's not as well predicted. And the reason for that is because these genes that are not well predicted are either gain of function, or there's also two genes where the pedigree are very complex, and actually these genes do not have a Mendelian inheritance pattern and are not strictly dominant. Finally, I wanted to finish with another interesting anecdote, which is about a gene that's called TCF3. This gene underlie a very severe autosomal dominant disease, E47 transcription factor deficiency, where affected individuals develop very severe infection in the first years of life. It still has a CONS of 0.41, meaning that it's under less negative selection than a random gene on the autosomes, where the mean is 0, right? So we wanted to see why, and it turns out that actually this gene so the autosomal dominant form of the disease has been found in 4 patients, but they all are mutated in the same domain of the gene. And other domain underlie a recessive disease that's milder. So what we found out is that using subaris, which is a domain-based version of negative selection, that actually this gene is not under selection for most of the domains, right? The value of subaris is higher than 80, meaning that more than 80% of the gene more than 80% of the gene domains are under stronger selection. But this small BHLH domain is under very strong selection with a value less than 20, meaning that only 20% of the domains are under stronger selection than this domain. So to conclude on the effect of negative selection on gene underlying inborn error of immunity, so I showed you that the gene that underlie dominant disease are under stronger selection than random gene, and that gene that underlie recessive disease. The loss of function genes are under stronger selection than the gain of function. The one that are within the loss of function, the one that are insufficient had stronger selection than the one that underlie negative dominant disease, but that it's also can be not uniform within a single gene. And I introduced two new scores, CONS, which is an unsupervised score, and SCONS, which is a supervised version that predict if a gene is likely to underlie a recessive or dominant disease. I want to thank my collaborator in Paris, and especially Laurent Abel, Louis Quintana-Mercy, and Etienne Patin that contributed greatly to this study, as well as Jean-Laurent Casanova in New York and Joe Gleason on the West Coast, and Anne Gregor that helped for the inborn neurodevelopment, as well as the different funding source. And I want to tell you that the previous version of this paper, based on the previous classification of inborn error of immunity, is available on bioRxiv, and that you can always contact me on Twitter if you have any questions.
Thank you, Franck. Thank you.
Thank you, Franck. So we have some questions. First to Julia and Marcus. How can a clinician recently obtain access to the PID HBO? Either one can reply.
I have to say, I think it's mostly to Marcus because I don't know how to access the ESID registry, for example. But if you just want to know the PID HBO phenotypes of just PIDs in general, it's just you go to the HBO website or you Google HBO, you find the website, first hit, click on it and write in the search bar the name of your favorite disease, click on it, and then you will get a list of HBO terms, if available, annotating the diseases.
All right, and then there are two questions to Andres. First, Mukina asks if GEMMADB is available in the App Store or somewhere else. That is a short question. And Marielle then asks whether this software that you use to build the GEMMADB would be open source so that other groups could copy it and then, when necessary, combine the data.
Yeah, so for the first question, no, it's not. As I said, it's a prototype that we built internally here at the CCI, and we hope that we can make it available, that you could just open your web browser and access it with a user and some login credentials. But for that, we need to go through the ethics votes and try to get to that point. So for the second point, the second question, yes, it is open source. It's MySQL and PHP, so all are open source languages. Of course, we could share the database structure or say some of the scripts, how it's built, how it's. I could send you the files, how it's done, but that would not be, I guess, the smartest approach, because probably what you and I and all the groups would benefit from is to combine all the data together. If we replicate the same at different points, it would be of no use. I mean, the idea is to try to, let's say, build databases such as OMIM, but specialized in immunology, where you have really the raw genetic data below. That you can compare, I could compare my patient with yours and so on. So, yeah, I mean, both options could be doable, but I personally think that the second one would be the most appropriate for the community and the scientific advancement. Sorry.
So we have a question for Frank. So as dominant negative effects are usually exerted by very specific mutations or clusters, do you think a more fine-grained domain-specific analysis may be useful, as shown for the TCF3 example?
Yes, thank you for this question. We do think so, yes. So we recommend to use, for now, the best tool that we have found to do that is Subaris, although, of course, we do hope that we'll be able to do a domain-specific version of Cons eventually that will solve this question. But yes, we exactly do expect that this would happen.
So there is another one. How to jump data to GEMMAP and use it? Do you think how to combine GEMMAP with the ESID registry project?
Yeah, so good question. So this is also what we thought and wanted to do. So we already discussed this with Gerhard Kindl, and that would be ideal. This, that would be our preferred solution. However, we don't know if it would be able, because of the different ethic votes that are required for the different projects. So if possible, we would like to do that. If not, probably we have to do a separate thing. But again, anyone could have access to it if we manage to open externally.
Okay, I have one question more to Julia and Marcus. Do you think it is worth the effort to implement HBO into the ESID registry, and how would you make those who enter data motivated to do that? Because it will anyhow be quite a bit of work.
So if I may answer, in my opinion, people could be motivated by the fact that they would automatically have their own prospective monitoring of the clinical courses of the patients and increase the output of information by facilitating the cross-referencing via across platforms. But of course, it is really a thing that will probably remain optional. So it has to be discussed and weighed if it's really worth it.
Thank you. I don't think there are any more questions. Or Anne, do you have any more?
I have plenty of questions, but I think we are short of time. So, I have one question. So you're saying that the recessive trait, the loss of function are under more selective pressure as compared to the gain of function. But that have a number of examples. So in the JAK-STAT pathway, for example, where gain of function have 100% of clinical phenotypes. So, and they are pretty severe. So even though it might be a bit later, a later onset in the severity of the disease, are you taking this into account, or?
So I think it's a problem of how we measure negative selection. And basically the reason is because negative selection is not really if a gene is constrained, if the sequence of a gene is constrained, but only if predicted deleterious mutation are less present than not. And for gain of function gene, most of them, but you have exception, only the gain of function mutation will be purged, but you may still have loss of function mutation that don't cause disease. Mm-hmm. And so because of that, this gene is going to be under less selection. It's still going to be constrained, but you won't see really this deleterious mutation being purged. So you won't find this gene under negative selection as much as you would expect.
Okay. So I think we have to close this session, correct? So thank you very much for attending the session, and thank you to the speaker for this great presentation. Nico, do you want to add something else?
No, I just want to thank the audience for participating. That's all.
Of course. Thank you, audience also. Thank you. Bye.
Thank you. Bye.
Bye.